The next section is entitled Cardiac Electrophysiology. Cardiac electrophysiology involves the use of electric currents to develop not only electric potentials inside individual cells, but also the electric signaling that coordinates a heartbeat. The cell membrane potential is what's responsible for the initiation of an action potential in a cardiac myocyte. The reason that cardiac cells are able to generate a membrane potential, or a change in the charge from the inside to the outside of the cell, is because they have a phospholipid bilayer. This phospholipid bilayer generates resistance between the inside and the outside of the cell and maintains a potential difference. This means that positive charges are shuttled outside of the cell and negative charges are kept inside the cell. Ions are able to flow through channels or transmembrane proteins that are specific for a certain type of ion and specific for a certain voltage or time during the cardiac cycle. It is the generation of the cell membrane potential or resting potential that allows cells to activate or generate an action potential. There are two types of cardiac action potentials when these cells are activated. One type is the fast response action potential, which is sodium channel dependent. This is present only in cardiac myocytes and in Purkinje fibers. The slow response action potential is calcium channel dependent, and this is only present in pacemaker cells in the sinoatrial and the atrioventricular node. These action potentials are able to spread from cell to cell because myocytes and pacemaker cells are connected to each other at gap junctions, which are located at the intercalated disc. So these cells are able to communicate directly with their neighbors, and ions are able to move freely through the gap junctions, so that an action potential in one cell will spread from it to its neighboring cell. It's the action potential that causes a cardiac cell to contract, and this is what causes the ventricle of the heart to squeeze. The myocardium, or the heart muscle, has two basic types of cells. Contractile cells, which are muscle cells, have a high amount of actin and myosin. These are atrial and ventricular tissues. They're the pumping cells of the heart. The contraction causes the shortening and the generation of pressure. Conducting cells, as well as pacemaker cells, are specialized muscle cells that do not contract. Their main job is to either form a pacemaker function or to generate a conduction that moves quickly down the wiring of the heart, also known as the Purkinje fibers or the bundle of His. The SA node is an example of a pacemaker. The sinoatrial cells do not contract, but they're able to generate a pacemaker potential, which causes the heart rhythm to occur. The atrial internodal tracts, also known as Bachmann's bundle, are also part of this conducting cell classification. They're similar to the Purkinje fibers in the ventricle. The AV node is another pacemaker of the cell that's located between the atrium and the ventricle. The bundle of His and the Purkinje system are fibers that, again, do not contract, but they do generate a signal that is able to travel quickly from the atrioventricular node to the myocytes in the ventricle. The cardiac conduction system is shown here. At point 1, at the roof of the right atrium, you have the sinoatrial node, that collection of pacemaker cells. They generate an action potential at a regular interval, usually approximately 60 to 100 beats per minute. That signal travels to the neighboring atrial myocytes through intercalated discs and causes contraction through the formation of action potentials in the right atrium. Bachmann's bundle carries the signal to the left atrium quickly so that the left atrium and the right atrium contract simultaneously. The signal then coalesces from all of the atrial myocytes at the atrioventricular node, point 2. The AV node is another pacemaker, but is usually overridden by the sinoatrial node's signal because the inherent rate of the atrioventricular node is slower than that of the sinoatrial node. The AV node then sends a signal down the bundle of His to point 3 and to the bundle branches, points 4 and 5, the right bundle and the left bundle. Also on this slide, you can see a diagram of a cardiac myocyte. The cardiac myocyte on top is seen to have multiple striations with multiple actin and myosin molecules that's responsible for contraction. At the bottom, you can see the difference between myocytes and Purkinje fibers. Purkinje fibers actually resemble neurons in that they have a very non-contractile appearing hollow interior, whereas myocytes are filled with actin and myosin. You can think of Purkinje fibers as wires, and you can think of myocytes as the engine of the heart. Again, the heart is composed of many different types of tissues, many different types of cells. The fast response action potential, which you see at the top of the screen, is seen in ventricular muscle and Purkinje cells. The slow response action potential, which has a slower slope, is seen in the sinoatrial node and the atrioventricular node. The fast response and slow response we're talking about here refers to the action potential upslope, not the automaticity. Remember that the sinoatrial node has the fastest automaticity in the entire heart. That's why it serves as the normal pacemaker of the heart. Resting membrane potential is the baseline voltage difference or potential across the phospholipid bilayer in the resting state. It's determined by the concentration of ions and the difference between those concentrations on the inside and the outside of the cell, as well as the permeability of the phospholipid bilayer for each ion. In fast fibers, such as ventricular myocytes and atrial myocytes, the resting membrane potential is negative 90 millivolts. That means that the interior component of the cell is more negative than the outside, and the difference is 90 millivolts. For the slow fibers, or the SA node and the AV node, those cells have an interior voltage of negative 60. In both cases, the interior is negative compared to the exterior. This negativity is generated mostly by the sodium-potassium ATPase. This slide summarizes the resting membrane potentials, the threshold potentials, and the currents as well as the conduction velocities of fast response tissues and slow response tissues. You can see that fast response tissues have a faster velocity, as you can see at the bottom of the screen. They have a higher amplitude. They have a higher slope. They have a more rapid current that's sodium dependent, not calcium dependent. And they have a lower threshold potential as well. The resting membrane potential is determined by the concentration gradient as well as the permeability. Listed here are the ionic gradients at resting in a cardiac myocyte. The interior potassium concentration is 140 millimolars. The exterior potassium concentration is 4 millimolars. And you can see the same for sodium and calcium. The permeability at resting, however, of the myocyte for potassium is much greater than that of sodium or calcium. Therefore, it is the potassium gradient that primarily sets up the resting potential. It's potassium that's responsible for the generation of the resting potential in cardiac myocytes. The inward rectifier channel is responsible for the leak of potassium, meaning the movement of potassium from the inside of the cell to the outside of the cell. Potassium is constantly being pumped into the cell by the sodium-potassium ATPase. The fact that it's able to move back outside the cell through these inward rectifier channels allows the performance or the administration of the resting membrane potential. Changes in potassium concentration can cause a change in the resting membrane potential. Hyperkalemia causes an increased extracellular potassium. This will reduce the concentration gradient. Hyperkalemia has been shown to prolong the QRS complex and hyperkalemia can also occur during cardiac ischemia. Acetylcholine also is able to increase potassium permeability. This creates a more negative membrane potential, leading to hyperpolarization. Hyperpolarization, or a more negative resting membrane potential, decreases automaticity, slows down heart rate, and decreases inotropy. Hypokalemia, or decreased extracellular potassium, may lead to torsade de pointe and ventricular tachycardia because of instability of the ventricular myocytes. The cardiac myocyte action potential is composed of 5 phases. The membrane potential decreases to the threshold, and then the action potential is generated. Phase 4 is the resting membrane potential. Phase 0 is the rapid upstroke. The rapid upstroke only occurs when the cardiac myocyte leaves its resting potential and hits the threshold potential. The threshold potential is approximately -70. This occurs when a neighboring cardiac myocyte depolarizes and the calcium ions from that cell enter the neighboring cell. The neighboring cell then is able to reach threshold potential and voltage-gated sodium channels open. The duration of the action potential varies based on the heart rate and the temperature. For example, hypothermia can prolong the action potential. The phase 0 of the action potential occurs just after threshold potential is reached, again usually from a neighboring cardiac myocyte through a gap junction. The upstroke of the action potential, or phase 0, is caused by activation of voltage-gated sodium channels. This causes an increase in sodium conductance, meaning the ability of the sodium to cross the lipid bilayer, and the current. The cell will then depolarize from -90 to +20 millivolts, meaning the inside of the cell becomes more positive. Sodium wants to move into the cell because of a concentration and a voltage gradient. At the beginning of phase 1, the internal component of the cell is more positive. The sodium channels inactivate, and voltage-gated potassium channels, called ITO channels, activate. This causes potassium to leave the cell. Potassium wants to leave the cell both because the internal part of the cell is positive and the external part of the cell has less potassium. This causes the external component of the cell to become more positive. This is why phase 1 is downsloping. In phase 2 of the action potential, we have what's called the plateau phase. This generally occurs at a voltage of 0, meaning the internal and the external parts of the cell are generally equal. The membrane potential at this point is 0, approximately. This is the longest phase of the cardiac action potential. It's caused by the opening of slow L-type calcium channels. The L stands for long, long-acting. This causes calcium influx into the cell. The ITO potassium channels then close, but the IKUR and IKR channels open. The UR and R stand for ultra-rapid and rapid. These channels allow potassium to leave the cell. Because calcium is entering the cell and potassium is leaving the cell, you get a plateau in the voltage. You have positive charges entering and positive charges leaving. The calcium enters the cell, and at this point the calcium is able to bind to troponin. So during phase 2 is when you see cardiac contraction because of calcium entry. Phase 3 of the action potential is the final repolarization of the cell. Those L-type calcium channels will close, but you have continued activation of the delayed rectifier potassium channels and the addition of the IKS channels. The S stands for slow. The inward rectifier channel, or IK1, will also open. The effect is potassium is pumped out of the cell, and the internal part of the cell becomes more negative. The cell then returns to the resting membrane potential. Phase 4 is resting potential. And at this point, the L-type calcium channels are completely inactivated, and IK1 channels, also known as inward rectifier channels, are activated. These are responsible for removing potassium from the inside of the cell. These are also responsible, as we mentioned before, for the initiation of the resting potential. At resting, potassium is moving in at the same rate as moving out. This is why the resting membrane potential is dependent on potassium. A cell's excitability is its ability to respond to stimulation. The resting membrane potential is the primary determinant of excitability. During an action potential, there are two periods called the effective refractory period and the relative refractory period. During the effective refractory period, another action potential cannot be produced. During the relative refractory period, a larger stimulus is required for the generation of another action potential. The pacemaker action potential of the heart is only seen in the SA and the AV node. The previous action potentials that we were looking at are only found in ventricular myocytes, Purkinje fibers, and in atrial myocytes. In the pacemaker action potential, phase 4 is upsloping. Remember, in the previous action potentials, phase 4 was mostly flat. The upsloping phase 4 is caused by a funny current. The funny current, which we will discuss later, is responsible for the pacemaker activity of the heart. Phase 0, instead of being related to sodium influx, is actually based on calcium influx in the pacemaker cells of the heart. There is no phase 2 plateau in the pacemaker action potential, and phase 3 repolarization is similar to that in the ventricular muscle. The funny current is what's responsible for the phase 4 depolarization in pacemaker cells. It's also present, but is much weaker in Purkinje cells and in myocytes. The funny current is what makes a pacemaker cell a pacemaker cell. It has recently been shown to consist of both inward sodium and potassium currents through channels called HCN channels. And HCN channels are activated by both hyperpolarization and by cyclic AMP, which is generated through adrenergic signaling through the sympathetic nervous system. HCN channels are inhibited by acetylcholine through the vagus nerve. Therefore, the funny current, the more upsloping it is, causes a faster heart rate, and this can be caused by adrenergic signaling or by hyperpolarization. The cardiac conduction system is initiated at the SA node. Sympathetic input to the sinoatrial node can increase heart rate. This is mostly carried out through epinephrine. Parasympathetic tone will decrease heart rate. This is mostly carried out through the vagus nerve. The effect of epinephrine and the vagus nerve on the funny current is what changes the heart rate. The signal that starts at the SA node propagates from cell to cell, from the right atrium to the left atrium. The signal then coalesces at the AV node and stops. The AV node is a slower conducting action potential because it relies on calcium. Patients with Wolff-Parkinson-White syndrome are also able to send a signal from the atrium directly to the ventricles through an accessory bundle or a bundle of Kent. When the AV node receives a signal from the atria, there is a pause. This pause allows for filling of the ventricle. Remember that after the atrium receives a signal from the SA node, it contracts, and blood moves from the atrium to the ventricle. In order to allow full filling of the ventricle, there is a pause at the AV node. Because the AV node requires calcium for its action potential, it moves more slowly than the cardiac myocytes. The AV node also has sympathetic and parasympathetic input. Therefore, dromotropy, or the speed of signaling through the AV node, is dependent on adrenergic signaling and vagal signaling as well. The funny current in the AV node determines the speed at which the AV node sends a signal to the ventricles. Once the AV node fires, the signal moves into the His bundle through the Purkinje fibers. This His bundle divides into the right bundle branch and left bundle branch, which are also made of Purkinje fibers. The left bundle divides into the anterior and posterior fascicles. This impulse terminates at Purkinje fibers and results in ventricular myocardial depolarization, because the ventricular myocytes are directly coupled to Purkinje fibers at their termination. This causes depolarization and ventricular contraction. In this diagram, you can see control pacemaker potentials with a funny current, an upslope, and a repolarization in red. The same pacemaker with parasympathetic stimulation, such as vagal input, causes prolonged slope as well as a decrease in resting membrane potential. This is in blue. This illustrates how parasympathetic input slows down heart rate because there are fewer action potentials per minute in the pacemaker that's received parasympathetic stimulation. Diagram B shows a sympathetic stimulation. Control again is in red. In green, you have sympathetic stimulation from epinephrine. And you can see that the heart rate has increased because the slope of the funny current has also increased. Cardiac mechanics are directly coupled to excitation. We call this excitation-contraction coupling. The actin-myosin cross-bridging requires calcium, and calcium is only present in the cytoplasm of the cell during phase 2, or the plateau of the action potential. Therefore, once calcium is present in the cytosol, this allows for actin-myosin binding and for contraction of the cardiac myocyte. The impulse from the sinoatrial node causes cellular depolarization. That cellular depolarization travels from cell to cell. The electrical event during the action potential is converted into a mechanical event called contraction. The mechanism of contraction depends on the transport of calcium ions into the cell during the phase 2 of the action potential. The calcium inside the cell also activates the sarcoplasmic reticulum of the cell, which is called calcium-induced calcium release, which further increases the amount of calcium in the cytoplasm of the cell. This calcium binds to troponin and allows for actin and myosin cross-bridging, which allows for the contraction of the cardiac cell. At the end of phase 2, however, the calcium influx ends. That calcium is either pumped out of the cell or it is pumped into the sarcoplasmic reticulum, a storage compartment inside the cardiac cell, which is similar to an endoplasmic reticulum in a normal cell. The sodium-calcium exchanger is a protein located both on the external surface of the cell as well as on the sarcoplasmic reticulum. The sodium on the outside of the cell pumps into the cell as calcium is pumped out of the cell. Why is it important for calcium to be released from the cytosol? This allows for relaxation. If the cell is not able to relax, then the ventricle is not able to relax, and diastole cannot occur. Therefore, it's just as important for calcium to leave the cytosol as it is for calcium to enter the cytosol. The intracellular calcium levels then drop because of calcium pumps, and this allows for relaxation and ventricular filling during diastole. During phase 2 of the action potential, not all troponin has calcium bound. This is the mechanism for increased contractility. As the cell increases the amount of calcium it has per action potential, the strength of the cardiac contraction increases. This is carried out through catecholamines, such as epinephrine and norepinephrine. These increase the amount of calcium that's taken up per action potential and increase the amount of calcium taken up by the sarcoplasmic reticulum. The more calcium is present in the sarcoplasmic reticulum, the more will be readily available for the next action potential. Another mechanism of increased contractility is cardiac glycosides, such as digoxin. Digoxin inhibits the sodium-potassium ATPase and therefore increases the amount of calcium that's stored in the sarcoplasmic reticulum. The increased amount of calcium binding to troponin through increased catecholamines and digoxin causes positive inotropy, or increased strength of contraction. Phospholamban, or PLB, is a molecule that rests on the sarcoplasmic reticulum. Normally, it inhibits the sarcoplasmic pump, the SERCA. The SERCA is responsible for pumping calcium into the sarcoplasmic reticulum. Phospholamban, when it's activated, decreases the uptake of sarcoplasmic reticulum, which decreases contractility, and it slows relaxation time. Phospholamban generally slows down the heart rate. Beta agonists, however, activate protein kinase A, which is cyclic AMP-dependent protein kinase. This enzyme phosphorylates phospholamban and inhibits it. When phospholamban is inhibited, this leads to more calcium uptake into the sarcoplasmic reticulum, causes higher contractility and faster heart rate. Basically, phospholamban is anti-inotropy, and beta agonists work by inhibiting phospholamban to increase calcium uptake into the sarcoplasmic reticulum and causing a higher contractility. The ryanodine receptor is another important molecule responsible for calcium-induced calcium release in cardiac muscle. When calcium is present in the cardiac cytosol, it binds to the ryanodine receptor. The ryanodine receptor then allows for the release of calcium from the sarcoplasmic reticulum into the cytosol. The ryanodine receptor is located on the sarcoplasmic reticulum. This is important clinically because ryanodine receptor mutations have been linked to malignant hyperthermia. Dantrolene, which is the cure for malignant hyperthermia, is actually a ryanodine receptor antagonist, whereas caffeine is a ryanodine receptor sensitizer, meaning it increases the sensitivity of the ryanodine receptor for calcium and increases the amount of calcium that's released in response to calcium. Remember that the ryanodine receptor is responsible for calcium-induced calcium release. This cartoon shows the relationship of phospholamban, the SERCA pump, and the ryanodine receptor to each other. It also shows the effect of catecholamines, such as epinephrine, on the beta receptor, increasing the amount of cyclic AMP. It also shows the effect of the action potential on the increase in calcium in the cell, and it shows how the cell is able to remove calcium through calcium pumps, the sodium-calcium exchanger, and it also shows the effect of cardiac glycosides on the sodium-potassium ATPase.